Additional possibilities in emerging approaches. Conventional fluorescence approaches rely on genetically encoded fluorescent probes that exploit fluoroproteins, mainly GFP, as the fluorophore and either periplasmic binding proteins or GPCR as a target binding component. Such probes traditionally rely on thread mechanisms where binding of a target changes the distance between a chromophore and a nearby cancer, thereby modulating the fluorescence efficiency. Most fluoroproteins include a rich protein structure that protects the chromophore, with flexible regions for attaching target binding receptors generally located far from the chromophore. These features restrict the movement of the pincer and chromophore upon target binding and result in probes with limited dynamic range and inability to detect analytes at low concentrations. Naturally permitted fluoroproteins overcome these challenges. In CPFPs, the original N and C terminals are fused using a peptide linker, while new terminals are formed near the chromophore. Such a structure imparts greater mobility to the fluoroproteins than that of the native variant, allowing greater reliability of the spectral characteristics. This is an example of the Gpcr-based intensiometric biosensor. CpEGFP is inserted into the third intracellular loop of Gpcr within the VFTD domain at the N-terminus. After binding neurotransmitter, the conformation of Gpcr changes that result in enhancement of CpEGFP fluorescence. Recent efforts expand the use of CPFPs to include detection of neurotransmitters. This is two-photon microscope image of a fluorescence hotspot associated with endogenous dopamine release. A family of dopamine-sensitive probes utilize CpGFP incorporated in the third loop of human B1 receptor to yield a positive feedback fluorescence probe. And in vivo studies in mice during dopamine transient induced by Q-reward learning tests illustrate the practical utility of these approaches as evaluated using conventional optical fibers. This is a working principle of a negative feedback near-infrared fluorescence sensor for detecting calcium ion, where the fluorescence efficiency decreases upon calcium ion binding. A calcium-sensitive probe results from insertion of calmodulin Rs20 into a biliverdin-binding monomeric infrared fluoroproteins. Here, binding changes the chromophore environment and decreases its fluorescence intensity. Looking at the wide-field fluorescence image, all biliverdin-binding fluoroproteins distributed evenly within the cytosol and nucleus of neurons with no apparent localized accumulation. This figure shows that co-expression of these three proteins enabled three-color in vitro imaging of spontaneous neuronal activity. Like this, the system designed for calcium detection is an example in which the advantages of CPFP over the thread-based approach are well demonstrated.
Now, the electrochemical Eptomer-based biosensor, which is a convenient approach to measure concentration of specific molecules directly in blood, even in situ in the living body, and feedback control drug administration. Here are the features of the sensors. Actually, this is a continuous measurement of a specific drug directly in the living body in real time. This illustration depicts the working principle of an Eptomer-based electrochemical sensor in which analyte binding induces a conformational change, which is observed as an increase in electron transfer to the electrode windows. This sensor uses this conformational change to generate an easily measurable electrochemical signal without any chemical transformation. To achieve this signal transduction, here the binding induced folding of an electrode bound Redox reporter, here it is methylene blue, modified aptamer leads to a change in an electron transfer that is easily detected using a square wave voltammeter. Here is the placement of the sensor in the mouse body. Oh, a cute rat. Well, this is cocaine. Cocaine has capacity to stimulate the central nervous system. A study says that within just 2 hours of single dose of cocaine, the mice had robust and quick growth of dendritic spines, but it's difficult to fully understand the mechanism of cocaine action. There are challenges in terms of real-time in vivo cocaine detection. An electrochemical Eptomer-based in vivo cocaine sensor on a silicon-based neural recording platform is capable to measure directly cocaine from discrete brain locations using square wave voltammetry. This inset shows a 40 magnified light microscope image of two unfunctionalized gold electrode sites separated by 200 mm. Dendritic gold deposition enhances the electrochemical signal that is understood from this scanning electron microscope image at 1000 magnification, that of a representative bare gold electrode site. Dendritic gold-coated electrode sites exhibit increased the peak current compared to bare electrode, indicating an increase in immobilized eptomer on dendritic coated electrodes. This is the definition of a square wave voltammetry. Experiments of the intravenous administration of cocaine were conducted to test the possibility of the cocaine sensor to measure physiologically temporary cocaine activity. This is the amount of cocaine concentration which decreases exponentially over a period of 45 minutes. This is the first direct measurement of physiologically relevant transient cocaine concentration in the brain. Finally, the conclusion. This latest review paper covers different types of modern and effective technologies in the field of neuroscience and explain briefly their features, explanation, and challenges. This is all about our presentation. Thank you.